friends, welcome back to my channel. Learners are here. I am with the next video regarding our topic DNA as a genetic material. So in our previous topic we learned about the Griffith's experiment, in which he told us about the transforming principle. But now in this experiment, like every McLeod and McCarty experiment, we are going to know what was the transforming principle in Griffith's experiment. It's a mixed effort, guys, of three scientists, namely Oswald Avery, Colin McLeod and Macklin McCarty. These three scientists, they carried out a series of experiments to identify the transforming principle. So the experiment consists of various parts. In the first step of this experiment, guys, scientists, they've prepared a purified extract of this S strain. Now, which S strain we are talking about? We are talking about the S strain cells found in this last mouse of the Griffith's experiment. In this last mouse, the mouse was injected with this heat-killed S strain bacteria and R strain mixture. And we got to know that in this mouse, transformation was found, because of which the mouse died. So they took these cells of this mouse and they prepared a purified solution of this S cells. So as we know that the cells contain different cellular components like DNA, RNA, proteins, etc. Here we are only talking about these three components. To dividing it into different parts, the solution was each time treated with different hydrolytic enzymes. Now guys, here the question arises, what does this hydrolytic enzyme means? Hydrolytic enzymes are different enzymes which cleave the substances or substrates in the presence of water, as the name suggests, hydro. So they act like scissors for these biochemical substances. Like for example, if we take a protein, as we know that protein is made up of long polypeptide chains of amino acid. So if we want to know the sequence, or if we want to cut these long polypeptide chains into small sequences, so we need a scissor. So here, as this is a biological component, guide, so we need a biochemical scissors. So this biological scissor here used for protein hydrolysis will be protease enzyme. This enzyme cleaves the protein chains. So as this solution will be treated with the protease enzyme, the protein in this solution will be degraded. Similarly here, the RNAs will degrade the RNA, and here the DNAs will degrade the DNA. In this solution, as we know that there is the absence of protein, but there are different other cellular components which are present in this test tube solution. Now to check or to get the results of this experiment, scientists have injected now these solutions into different mouse to get the results. Now in the first case, when the mouse was injected with the protease treated solution, then the mouse died. Similarly, in the same happened with the RNase treated solution. But unfortunately, what happened in the third case, the mouse, when it was injected with DNase treated solution, the mouse lived. Now why this happened? So to know the answer to this question, why did mouse lived, we come back to our previous video, a part of that this mouse was containing the heat-killed S strain and R strain. And we came to know that in the last mouse of the Griffith's experiment, the last mouse of the Griffith's experiment mouse died because of the transformation that occurred in this mouse. And then Griffith gave the hypothesis of the transforming principle, that something has transferred from the S strain, heat-killed S strain, to the R strain, which has transformed these R strain into the violent S strain. So here also the same thing occurs. Here if we are talking, So in the first case, the mouse died because of the transformation that has occurred in the mouse. Similarly, in the second case, also with the mouse died because here also the transformation occurred and the R strain, the non-violent R strain were converted to violent S strain cells. But in this case, what happened, guys? Mouse lived because this proves that no transformation occurred. Now, the reason why not the transformation occurred was that DNA got degraded. Here, the degradation of DNA with the DNase proves that DNA was degraded, and hence DNA was the material which has been transferred from the heat-killed S strain to the R strain, which has transformed the R strain cells. And we came to know that what was the transforming principle. So what was the transforming principle, guys? Here the transforming principle was the DNA. So this is the end of this part of the experiment, guys. So in my next video, I will come with the third and the final part of this topic, discovery of the genetic material. So till then, bye bye, and don't forget to like and subscribe my channel. Thank you, guys.